Onto the front cover, we popped the front panel cover off and then used a Dremel to mill a slot. We also drilled a hole for the power switch. For the Arduino, and also for the solenoid, we 3D printed a custom box and a custom mount to make it easy to mount them to the back cavity using just a couple screws. With the main electronics mounted and wires run, things can just be plugged in, and the tubing connected. In terms of electronics, we had to make a custom shield to fit on the Arduino with all the different bits and bobs. The schematic, of course, is in the description. Since the point of the incubator is to maintain a specific CO2 concentration, we want to minimize leaks, both so we aren't wasting CO2 and so normal air can get in. So anything that was in the main chamber of the incubator was sealed in with mercury seal. The only other thing we changed was the color of the light that's built into the incubator by default. Normally it's blue, but we subbed it out with some white LEDs. Since most mammalian media has a color indicator in it so you can see if there's a problem at a glance, it's really important to be able to see that color accurately. But with that, the whole thing can be closed up, and all that's needed is a nice vinyl decal to make it complete. Alright, enough messing around. It's mouse meat time. Like pretty much anything you want to grow, mouse cells need food, and that comes in the form of a special culture media. The media we're going to be using is called DMAM, or Dalbeco's Modified Eagle Medium, which, as I've said before, disappointingly doesn't contain eagles. But it does contain almost everything else that a growing cell needs. As a quick test of the incubator, just to make sure that everything's working, we loaded some plain DMAM into a 6-well plate, and then stuck that in the incubator and let it come up to 5%. Here's what the plate looks like before it goes in, and here it is after it's been in the incubator for a while. The color change is kind of subtle, but it is distinct, and as someone that's done a lot of mammalian culture, you learn to recognize this beautiful red-orange color as the color of happy media. So the incubator is working properly. Now for the real deal. The cells we'll be growing are a primary cell line derived from adult mouse skin, specifically fibroblasts. We chose these because they grow rapidly, are non-human and unlikely to contain any pathogens, and don't require anything that particularly weird or expensive to grow. They're also